Hello students, welcome back. In the previous video, we gained knowledge about the structure of prokaryotic cells. Their cell structure is simple. In this video, let's learn about the structure of eukaryotes like protists, animals, plants and fungi. Their cytoplasm shows compartmentalization by the presence of membrane-bound cell organelles which prokaryotes lack. Eukaryotes have a well-defined nucleus and nuclear membrane. The presence of this nuclear membrane is what sets prokaryotes and eukaryotes apart. Let's look into the details of the cellular organelles in eukaryotes. The innovation of electron microscope aided in the detailed study of the cell membrane. Scientists could give more information about the plasma membrane by analyzing the RBCs. An improved model of plasma membrane was proposed by Singer and Nicholson in 1973, known as the fluid mosaic model of plasma membrane. According to this model, the membrane consists of phospholipids, proteins and cholesterol which contribute towards the flexible nature of the membrane. The cell membrane is flexible. It can move in a wave-like motion. The cell membrane mainly consists of lipids and proteins. It has a lipid bilayer, that means it has two layers of lipids. Here you can see this is the first layer of lipid. This is the first layer and this is the second layer of lipids. That means it is a lipid bilayer. These lipids are called phospholipids. Phospholipids have a hydrophilic head and a hydrophobic tail. Hydrophilic means water-loving. They get absorbed in water. This is due to the presence of a negatively charged phosphate group. Thus, the phosphate head attracted towards water. On the other hand, hydrophobic means water-resistant because they are non-polar. That means they do not have charge at the end. Water is polar. That is the reason hydrophobic tail do not interact with water. These are the hydrophilic heads of this layer. These hydrophilic heads face extracellular fluid, while the hydrophilic head of this layer faces the cytoplasm inside the cell. The hydrophobic tails are protected from aqueous environment and are directed towards each other. They are not exposed to water, either to the extracellular fluid or to the cell cytoplasm. In addition to the presence of phospholipids, the plasma membrane also contains cholesterol. Cholesterol regulates membrane fluidity by controlling the movement of cell membrane components. The cell membrane also possesses proteins and carbohydrates. In human beings, the membrane of RBC has 52% proteins and 40% lipids. Here you can see these are the carbohydrates. They are attached to the proteins in the cell membrane. There are mainly two types of proteins called integral proteins and peripheral proteins. Integral proteins are found throughout the cell membrane, while the peripheral proteins are attached to the top or periphery of the cell membrane or to the surface of the integral proteins. It can be found here as well, or to the internal surface of the cell membrane that faces towards the cytoplasm. These peripheral proteins can be attached and detached from the cell, but integral proteins are permanently attached to the membrane. Some integral proteins are called channel proteins. They have a hole that allows certain ions to pass from the extracellular fluid to the cytoplasm. Polar molecules with a charge are repelled by the nonpolar hydrophobic tail of the phospholipids. This prevents them from entering the cell through the membrane. Consider sodium ions as an example. If the concentration of sodium ions are very less inside the cell, here you can see there are no sodium ions inside the cell, but the concentration of sodium ions are high outside the cell. So if the concentration of sodium ions are very less inside the cell, sodium ions from outside have to reach inside the cell. But as we know, the hydrophobic tail will not allow it to pass through the cell membrane. The role of channel proteins become crucial in this scenario. These ions can pass through the channel proteins into and outside the cell according to the concentration gradient. If the concentration of certain ions is high inside the cell, the channel proteins will let them pass to the outside because the concentration is low outside the cell compared to inside. The same is true in the opposite direction. Since the passage occurs along the concentration gradient, that is from higher concentration gradient to the lower concentration gradient, it does not require any energy or ATP. Another type of protein called carrier proteins. Carrier proteins have a remarkable ability to transport ions against their concentration gradient. That means even though there is high concentration of certain ions inside the cell and still the cell requires additional ions to perform a function, carrier proteins transport ions from the outside, that is where the concentration is low, to the inside of the cell against the concentration gradient. These channel proteins transport ions from outside of the cell to the inside against the concentration gradient, but it needs ATP as the passage is against the concentration gradient. Understanding the proteins in the cell membrane help us to grasp how particles move through it. The movement of molecules through the membrane is by the following means. There are two types of transport, passive transport and active transport. Let's learn about them. There are three types of transport in passive transport, that is diffusion, osmosis and facilitated diffusion. Molecules can enter the cell if its concentration is higher outside the cell and lower inside the cell. Diffusion is the movement of molecules across the cell membrane from an area of higher concentration to an area of lower concentration. The molecules will pass until the concentration outside and inside of the cell will be equal. For example, is the passage of oxygen and carbon dioxide. Oxygen and carbon dioxide enters the cell through diffusion. The transport of water molecules down the concentration gradient through the cell membrane is called osmosis. But due to the presence of hydrophobic tails of phospholipids, the passage of water through osmosis happens slowly. However, it can happen in a faster pace by facilitated diffusion. Let's learn about facilitated diffusion. Just a few minutes ago, we were introduced to the channel proteins which enable facilitated diffusion from regions of higher concentration to the regions of lower concentration. Facilitated diffusion allows fast entry of water molecules into the cell. This process also allows larger molecules that cannot enter through the cell membrane by simple diffusion to the cell. All these kinds of transport do not require energy or ATP, as this transportation happens from higher concentration gradient to lower concentration gradient. Now, let's learn about the active transport. This is the passage of molecules against the concentration gradient. That means the molecules pass from lower concentration gradient to higher concentration gradient. ATP is required for this process. Examples are the movement of glucose and amino acids which cells needs in larger quantities. If you like this video, please share it with your friends. If you have any queries, put it in the comment box. Thank you.